let's begin again. So I'm delighted to introduce Olivier Vonnecker, who is from Belgium. I met him quite a long time ago when I first was working with Anne de Pep, who was, I'm not sure I would say his teacher, but certainly a person who imparted a good deal of knowledge on PXE to at least me, and I think others. And then Olivier has been working for a very long time himself in PXE. And so without further ado, I'll turn it over to you, Olivier. We have an hour set aside, and we like to leave time for questions.
Okay, great. I'll just share my screen. There we go. See if that works. Okay, I just wanted to ask if you can see my pointer or not.
Yes, I can. And lots of people are low vision, so they won't be able to see it. So what we ask is that you describe whatever you're pointing to.
Okay. So good morning, good afternoon, good evening to you all. For me, it's good evening. But wherever you are, in the next 40-45 minutes, I will try to entertain you on the genetics behind Pseudoxanthoma elasticum, because as you may have read in the program, the original gene for PXE, which is ABCC6, has been described somewhat 20 years ago. But as you know, genetics is a rapidly evolving science, and if we just take a brief overview in time, as you will see here, we start in 1866, which is not that long ago, with Mendel, who described patterns of inheritance, how characteristics can be transmitted from one generation to another. It was discovered as a molecule in 1869, and it took over 100 years until 1953 to actually figure out how that DNA looked and what the structure was, the famous double helix structure of Watson and Crick. And since then, there have been some steps moving forward until 1990, when there was a big project which called Genome, that actually set out to analyze the complete genetic material of a single individual. And they took their time. It started in 1990 and it ended in 2000, when the genetic material was characterized of one single individual. But since then, we have really had an explosion of knowledge, possibilities, of insight in genetics, with explaining a lot, sometimes also giving new questions. And of course, in PXE, PXE is not an exception to the rule. This has been exactly the same evolution. And some of these things that have evolved are how we look at genetic causes of disease. Originally, we thought one disease, one gene, but now we know that there are multiple, can be multiple genes that lead to the same disease. Also, that diseases are not standing by themselves, but are part of a larger spectrum of disorders that look alike, but are not completely the same. And that besides the genes that actually cause the disease, that you can see that there are other genetic factors that will modify how an individual experiences his disease, whether it is more severe, whether it starts earlier, whether he has more this kind of symptoms or more another type of symptoms. And these genes are called modifier genes, because they modify the way that the disease presents itself. Based on that, we have been able not just to test a single gene, but also test all genes in one analysis, or even test the whole genetic material of an individual, not in 13 years, but approximately in a few hours. We can do diagnostic testing, we can test carriers, and one novel thing is expanded carrier screening, where couples that have a child wish but are not sick and they don't have a family history can get tested for a large set of genetic disorders to see whether they carry a genetic defect for them. And for example, in many of these test kits or panels that are offered, pseudoxanthomylasticum is one of these diseases. And we can also test embryos in a procedure that is called pre-implantation genetic diagnosis. This has led to a set of interventions, embryo selection, if we want to prevent a disease from being to the next generation, or pregnancy interruption. And also possibilities, mainly in research at this point, of targeting the genetic defects with types of therapy specific for a certain genetic defect or through genetic modification. And for many of these things, there are examples in PXE, and I'll walk you through this to explain this a little bit. There is a famous movie quote that says, "Assumption is the mother of all fuck-ups." But nevertheless, I am going to start with a big assumption. And that assumption is that some or many of you do not have a lot of, let's say, basic genetic knowledge on how your DNA works and how it is built. And so I'm going to start with a few slides, just hinting on some key points that you need to know to understand everything else. If you already are a biologist by training, for example, you can go get another sandwich or something. If you're not, then the next few slides are extremely important. And as you can see, the compound of our interest, or the molecule of our interest, is DNA, deoxyribonucleic acid. The name is not so important. And you can see the beautiful structure. It's a helix. There are two strands, approximately 3 meters long, that are interconnected with one another. And I thought a lot in many years ago on how to explain how DNA works, how it is built, to people that do not have a biology background. And usually I use the analogy that DNA is the music score of life. It guides you from the overture up to the finale, and it decides in many cases how and when you will reach that finale. And if you look at the music score, of course, you have lines where the music is written on, and you also have structures where your DNA is put on, and those are called the chromosomes. And there are 46 chromosomes that carry the DNA. Obviously, on a music score, you have notes that build the music itself, 7 notes. And in DNA, those are letters. Where are they? They're not coming.
They decided it's too late at night to be.
Yeah, apparently. Let's see, that works again. There are the notes, at least. So we already have music. Let's see if we also. Yeah, then we have the letters. 4 letters, A, C, G, and T, which are also called the full nucleotides. So you see there are only 4 letters. So music, genetics is really ridiculously simple. And these letters, it's really strange because I don't know what's might be why this is, but I'm going to show it like this then for this slide. These letters form a strand of about 3 billion of these letters all together with genetic code. And then you can see that there are, if you have a music score, there's not just the notes, there are other symbols on there that enrich the music. And this is also in your genetic material. Sometimes there is a change in a letter, a variation that does not cause disease, but just makes us all a bit different. And this is called a polymorphism or a neutral variant. And then finally, of course, everybody knows that, especially if you sing in the shower, the false notes that do not really improve the music, and that you can also have in DNA. You have certain variation that is not neutral, but leads to a disease, and that is called a mutation or a genetic defect. Let's try this again. So in this overview, your genetic material is in your cell. It is in your on your chromosomes, and if you pull at the chromosome, you get this 3-meter strand that actually consists of all these 3 billion letters A, C, G, and T. And everybody talks about a gene, and actually a gene is just a piece of that DNA, a piece of that strand of letters that contains the instructions to make a protein, like here, the protein, let's say ABCC6, the main protein involved in PXE. As such, you have about 22,000 of these genes in your complete genetic material. Very important is that from each of the genes, you have 2 copies. And for the rest of the presentation, these copies, if they are normal, they are in blue. And you can have, of course, a genetic defect in one of them, then you have a blue one and a red one. Or you can have 2 genetic defects in each copy of the gene, then you have 2 red genes. If that is the case, obviously, the gene that contains the instructions will be abnormal. You will have the formation of an abnormal protein, leading to a less functional protein. It can still function a little bit, but it's not completely as it should be. Or worst case scenario, the protein is completely absent. And as a result of these 2 scenarios, you will get a disease. And in the case of ABCC6, that would be PXE. Now, these genes, 22,000, is only, although they are very important, only 2.5% of your genetic material, which is called the exome. And the rest, which is what is depicted here, the whole apple, is called the genome. And you can ask yourself, of course, if 2.5% is all your genes, what the hell does the rest do? And probably the rest is important to regulate these genes to make sure that they are active at the right moment, that they stop being active at the right moment, that they are active at the right percentage at the right moment in the right tissue, etc. Okay. Knowing that, you are full-learned geneticists, because this is basically everything that you need to know to understand genetics. It's not that difficult. So let's head back to PXE. You already had the introduction on all kinds of symptoms. I'm not going to go into that, but important to take home is, or to remember, later on, to find some explanations for this variability in type of lesions or the evolution of, and they all occur, of course, based on ectopic mineralization, this calcification of your elastic fibers in your connective tissue in different tissues. Now, if we look at the inheritance of PXE, most families that I encounter, or patients that I encounter, they have a family tree like this, where you have a single individual, which is marked in white here, but brothers, sisters, parents, everybody else is healthy. There's just one. Besides that, there are other families where you see that there are multiple affected patients in the same generation. So, for example, in this case, a brother and a sister marked in white who have the disease. The parents are healthy, and there's another healthy brother in the middle there. And this was very suggestive for an autosomal, what is called an autosomal recessive inheritance pattern, meaning that the affected patients have 2 genetic defects in each copy of their gene. And if you go back to the higher generation, to the parents, you would find that the parents each carry 1 genetic defect and also have a healthy copy of the gene, and hence do not develop a classic pseudoxanthoma elasticum. Strangely enough, there were also families where you saw that there was, in subsequent generations, a patient that was with PXE, for example, a parent and a child. And this was initially thought to be what is called autosomal dominant, being that you do not need 2 genetic defects to have the disease, but just 1 was enough and could be transmitted very easily from the parent to the child. This was further addressed and solved in 2000 when the actual gene was found, because before that this was simply hypothesis based on the families that were encountered. But in 2000, the ABCC6 gene was identified with this strange expression profile, though where the gene was active, not at all in the tissues where you actually see the symptoms, but rather in the liver and to a lesser extent in the kidneys. And giving rise to this very complex protein, a transporter protein, part of a very large family, which is still somewhat ambiguous, and it still surprises us even after 20 years that there are novel functions, unclear functions. We don't know what the substrate is, what it transports, and it works in a very complex set of mechanisms where, for example, the substance inorganic pyrophosphate, which is a calcification inhibitor, is very important. But as said, it is certainly not true that ABCC his secrets at the moment. But the ABCC6 gene did make clear how the inheritance of PXE works. It confirmed the autosomal recessive inheritance. So patients have two genetic defects inherited from their parents who carried each one genetic defect. Of course, genetics is the discipline of the exception to the rule. And in some cases, you can see, for example, that one of the parents does not carry a genetic defect. So the child has two genetic defects. In this case, the mother also has a genetic defect and the father does not have a genetic defect. This can happen because the patient himself makes a novel genetic defect. This is rare, but it can occur in recessive disease. Sometimes you see that if you do not find it in blood in the parent, that it is present in the egg cells of the mother, or in this case, in the sperm cells of the father. With a complex term, this is called germline mosaicism. But this is very important because this still poses a risk for carrier parents or parents of a child with PXE that want a novel or another child. There is still a risk of having a child with PXE, even if you do not find the genetic mutation in the blood of one of those parents. And certainly if it is a father, non-paternity is something that we and have to explain them. For the third category, so these apparently autosomal dominant ones, where you have PXE in the subsequent generations, it was found that this is not really true. If you looked at these patients, so the mother and the daughter in this case, they both had two genetic defects, but actually the mother married an individual who had a single genetic defect, so a heterozygous carrier who was healthy, obviously, who does not have PXE, because he had another normal copy. And they both transmitted one of their defects to, in this case, their daughter. And so it one generation to another, it is done, but it is based on two defects, and that's why it's called pseudodominance. Very in the beginning, ABCC6 made it clear that it would not make it easy for us because, you know, you have the gene, but there were certain parts of your genetic material in these patients that looked very much like the gene and made it very difficult to perform the analysis. And these kind of genetics parts of the genome that look very similar or are nearly identical to a certain gene are called pseudogenes, because they don't really produce a protein, as we saw in one of the first slides. And this is important because when you do a genetic analysis of the gene, you want to specifically look at the ABCC6 gene, and actually you do not want the pseudogenes. So we are really interested in one of in a genetic defect that lies in the ABCC6 gene, because we know that it causes disease. But if that same variant would be in one of the pseudogenes, it would be completely innocent. So we really have to know, if we do the analysis, are we looking at that actual ABCC6 gene or one of these pseudogenes? And so we needed to adapt our genetic analysis genes, because otherwise the genetic information that we would get would be completely false. Why are these pseudogenes there? Is that just to annoy the geneticists? Completely. These pseudogenes have a function regulating ABCC6. The mechanisms of that are not completely clear, but this is one of the things that it actually that it does. Okay. Knowing this and knowing the ABC, that in about 90% of patients we find 2 mutations, 5 to 7% we find 1 genetic defect, and we are missing the second one most probably. And in about 3 to 5% we do not find any mutations so far because we are missing it for technical reasons, or because we are looking at another gene that is the cause of the disease in those patients. These mutations are really scattered throughout the gene, or in this case, the protein that is shown with the colored dots are the mutations that are present. There are over 300 of them. So it is one of these diseases with a very large mutation spectrum compared to mutations that, or diseases that sometimes are caused by a single mutation or by 10 or 20 mutations. So here there are more than 300. There are some that are very frequent, but the majority are what are called missense variants. And this is not unique to PXE. This is the case in many genetic disorders. But they pose a certain challenge when we find these kind of variants. So I just want to highlight these different types of sequence variation. Silent variants, missense variants, nonsense variants, frameshift variants. I'm not going to go into detail, but in a missense variant is just usually one letter of your genetic code that is different, and that will result in, if you have a normal protein, that will result in a protein that is less functional. So the protein will usually still be there, but it will function a little bit less than you would expect, and that can lead to a disease. This is different from a nonsense and a frameshift variant, because in these cases you will usually see that there is absolutely no protein left anymore. And this category of missense variants has, let's say, in the last 3 to 5 years, has put us in another problem, and that is variant interpretation. And that means that when I started doing sequencing of ABCC6, and that is 15, 16 years ago, I think that I first tried to sequence ABCC6 because it failed a lot in the beginning. We found a missense variant, and we very quickly said, okay, we find this in a PXE patient, it's a mutation. And we used like 200 control patients, and certainly if it was not present in those 200 controls, this was a good argument that this was a mutation. Now we use hundreds of thousands of controls because we have large databases of individuals who are healthy and their genetic data. So compared to that, those 200 that we used 15 years ago is completely crazy, but it was the best that we had at that time. And in many diseases, we see that what we originally thought was an actual genetic defect was not the case, and it was rather a neutral or an innocent polymorphism. And this was particularly true for missense variants, that this could be the case. So these missense variants can either be neutral, they can be disease-causing, but in both cases, they pretty much look the same when you look at the genetic material. It's just one letter that changes, and it's very difficult in some cases to find out what is it now. Is it normal variation, or is it really causing PXE? So there is a challenge in interpretation of the meaning of this kind of variants. And so we asked the question, are we sure that every missense that's out there reported as being disease-causing, that is actually the case or not? And these variants, genetic variants in general, can be classified in 5 classes. 1 and 2 those are, polymorphisms. These are innocent neutral variants. 4 and 5 are disease causing for sure. And then you have class 3, which is the middle, and those are variants of unknown significance. That's the worst thing that you can have as information as a patient. That is, we find something, so it's not completely normal, but we're not really sure whether it is actually disease causing. It can go either way. It can be disease causing, or it can be completely neutral, and we don't really know very well. And as you can see, if you look at the missense variants that are reported in ABCC6, there is a fairly large proportion, 60–70%, that are actually class 3 variants of unknown significance. We can clean that figure up a bit, make it a bit better if we say, okay, the variants that are very close to the border of class 4 and 5, so the variants that are actually disease causing, those are most probably also disease causing. So we can, increase that with 1/3 of missense variants that are in that situation. But overall, from the 286 missense variants that are out there as being disease causing, about half of them we can say for sure with the knowledge that we have today that they are disease causing, and the other half are questionable. They can be disease causing or they can be benign. And one of the reasons for that is, and this is also partially my fault or my responsibility, is that many of these variants have been detected 15 years ago and have been evaluated with the knowledge of 15 years ago. And at that time, everything pointed out towards the fact that they were disease causing, but knowledge evolves. And sometimes you have to correct certain of this information, or admit that with the current information that we can't be so sure anymore. Is this important? Well, of course, you can say, it's just 2 mutations. I already know that I have PXE because I have the symptoms, and I had a skin biopsy taken, and there is mineralization. So they have 2 variants, whether they are really causal or not. What are you fussing about? But nonetheless, if there is a clear-cut PXE, you don't really need the molecular confirmation. But in many cases, I see patients where you don't have a clear-cut clinical diagnosis. And then, of course, the molecular diagnosis is not just a confirmation, but it's really made or used to have a diagnosis. And if we are looking at the wrong variant, then we are giving that patient a wrong diagnosis. So for patient counseling, it's very important, but also for pre-symptomatic testing. Let's say that you have a family with, in this case, a woman who has PXE. As you can see, the parents are, and that woman also has a younger brother, and the brother apparently does not have any symptoms, but in theory, genetically seeing, he has 4 possibilities. He has inherited the 2 normal copies of, has inherited 1 genetic defect, then he's a carrier, or he has inherited the 2 genetic defects, and for example, does not have any visible skin lesions, and does not yet have any eye problems. And so in that case, we would use the genetic defects that we found in his sister to do pre-symptomatic testing in him. That, of course, if we would find the 2 genetic defects, we would say you will also get PXE, or you will also have PXE. But you can also only say that reliably if you are absolutely sure that the genetic defects in the sister are really disease causing. So for pre-symptomatic testing, you really have to be 100% sure that the genetic defects that you use are really disease causing. Otherwise, you will give this man a wrong diagnosis. The same is true for carrier detection. So we know that carriers of 1 genetic defect can have calcifications, which do not really lead to clinical problems. They see them on ultrasound in the liver, in the kidney, but they don't have kidney or liver problems. But they have a higher risk for periodontal disease in the legs. And for example, it has been shown that these kind of single mutations are much more frequent in patients that suffer a stroke. So we in families of PXE patients, and for example, because of the higher risk for stroke, in some cases we will do prevent. We see that there is a beginning of vascular disease. We will also put them, for example, on aspirin. But again, you will do those things if you detect a carrier, but you want to make sure that the genetic defect that you are looking at is the causal, because otherwise you will point out carriers based on a neutral polymorphism or instant variant. The same is true if parents carry a genetic defect. Each of the parents carries a genetic defect, has a 25% chance of a child with PXE, and once embryo selection, so pre-implantation genetic diagnosis. That may be a bit of a difference, but in Europe we can do this if parents want this. We have already done this, but again, we will only select embryos if we are absolutely sure that the genetic defect is really disease causing. If there is any question about causality, we will not step into this procedure. So for a class 3 variant of unknown significance, this cannot be done. And finally, there are experiments with gene therapy, again, mostly in research. It seems to work in animals, but fortunately, humans are not animals. Careful with messing with your genetic material, and certainly also the side effects of this. But even when they would move to clinical practice, knowing your genetic defects, your mutations will be absolutely vital. And of course, knowing that they are absolutely disease-causing, that there is no doubt about that. Which is why we need to evaluate these missense variants. And we can use different approaches for that. One of them is using Zebrafish. I'm not going to go into that today, but tomorrow during the research session, I will give a very short speech on why fish are so interesting for a disease that they have affinity with. So you will have to tune in tomorrow for that if you want to hear it. Just focusing on the fact that, as I said, we find in 90% 2 mutations, but in 3 to 5% of patients, we do not find any mutation. And this has led to the fact that over the years, we have seen that, you know, you have PXE, but you have multiple genes that are involved in PXE, not just ABCC6. And PXE is actually part of a spectrum of diseases. And one of the first ones that was the PXE-like disease with a coagulation deficiency, with a very severe skin phenotype due to mutations in a completely different gene called GGCX. And since then,
I'm just
there have been other phenotypes. Sorry?
Can I interrupt you just 1 second? Because Yoni showed this PXE-like photograph as extreme PXE, and I just want to make the point here that this is not PXE the way we traditionally think about it, and that people don't have to worry this is going to happen to their abdomen and their whole arm or any of that stuff.
No,
Great.
No, exactly. So this is I'll go back again. So no, go back. Yeah, so this one is a different disease. That's the whole idea. It's part of a disease spectrum. So this is another gene, GGCX, and as you can see, it's not something that you see in PXE. The texture of the skin is also completely different. It's very thick. I mean, if I have to take a blood sample in that patient, I really have to push hard against the skin. It's completely different than in PXE, but it started. And the same is true for this one. Again, mutations in GGCX. They also have a retinopathy, but the skin features are different. compared to PXE. But if you take a skin biopsy, it looks nearly the same. And so this GGCX gene is important in a metabolic cycle in your body, which recycles vitamin K. Vitamin K is not very much present in your food, so your body has to recycle it. And this GGCX is crucial for that. And by doing that, it activates certain calcification inhibitors. So if that GGCX gene contains a genetic defect, these calcification inhibitors will not be activated anymore, and you will get calcification based on that. And then it was also seen that sometimes you can have an overlap. You can have a patient who has one genetic defect in ABCC6 and another genetic defect in GGCX. So two genes that are actually involved in developing a phenotype, a set of symptoms that looks like PXE and looks a bit like the PXE-like disease with coagulation deficiency. So really a mixture of the two. And then finally, it was also seen that a third gene called ENPP1 was also involved in PXE. And this gene has been a long time known to cause a very severe calcification disorder, which is called GACI, stands for Generalized Arterial Calcification of Infancy. These are babies that have terrible calcification of their blood vessels, which is life-threatening. But in some cases, when they survive, they develop symptoms that look very much like PXE. And it has been shown that classic PXE patients, in rare cases, can have mutations also in ENPP1 and vice versa. Some cases, some children with this arterial vascular calcification can have mutations in ABCC6. So these two diseases, although they are completely different, PXE onset, middle puberty up to adult onset, and very slowly progressive. GACI is very often already in the womb that you can see the calcification in the baby, and is very acute, very rapidly. Different diseases, but the same genes are involved. And this ENPP1 gene plays an important role in the processing of that other calcification inhibitor that I told you about in the beginning, inorganic pyrophosphate. So multiple genes that cause the same disease and variants that are difficult to interpret in some cases, and some cases. That all why we need geneticists? And the answer is no, of course not. Because if you look at again, these different variants, and specifically the missense variants and the nonsense variants, you can actually have, you can have different combinations. You have 2 times. You can have 2 missense variants, which means that you'd have 2 times a protein that function a little bit less. You can have a missense and a nonsense, which would mean that you have a protein that functions a little bit less and then a completely absent protein. And then, for example, 2 nonsense mutations where you would have absolutely no protein left. So you would think that the patients in the latter category have the worst disease because they don't have any protein left. And the answer to that is no, because there is no clear association between mutations and symptoms. You cannot predict the symptoms, the cause of the disease, silent the aggressivity of the disease based on the mutations that you find in, for example, ABCC6. And this was further seen because there are mutations in ABCC6. There are 2 here. And in one patient, they cause pseudoxanthoma elasticum. And these identical mutations in ABCC6, in another patient, they cause that GACI phenotype, that vascular phenotype. So in one patient, they cause a very chronic, slowly progressive disease, and in the other patient, the same mutations cause a very acute, aggressive, and early onset disease. And the question is, what is going on here? It's clearly not only those 2 mutations that decide which phenotype that a patient gets. There are a lot of reasons to explain disease variability. Environment, obviously, if you smoke, if you have PXE and you smoke, that is a terrible combination. And you will get vascular disease for sure, and it will aggravate the vascular disease. So this is an environmental factor. But there are also genetic factors that cause that. And those are those famous modifier genes, genes that do not cause the disease, but they modify the course of the disease. They make it more severe, or in some cases, they make it less severe. You can have different kinds of modifier genes. Some of them are very powerful. This is the thick, powerful effect, and in themselves, one gene can actually steer a part of the disease, make it more severe, make it milder, etc. But in many cases, these modifier genes only have a very small effect when you look at them individually. But if they all work together in a single individual, they can push the disease or part of the disease into a certain direction, make it milder or make it more aggressive. And again, for PXE, modifier genes is something that has been or is being looked for very actively. It's very difficult. And if you look at lists of for many diseases, there are genes. And if you look at humans with the same disease, you will see that that list is extremely short because they are very difficult to find and to validate. But there are some modifier genes that are known, large effect modifier genes to some extent. You can see them here, and I will just give you an example of VEGF-A and of ENPP1 in a minute. And then we are also looking very actively at small effect modifier genes, and we have come up with some good candidates, which also need to be validated further, so they can't really be used, these small effect modifier genes, in the clinic yet. But we hope that in the next couple of years, of course, that we will gain more knowledge that would enable us to predict how the disease will evolve. Just as an example of a large modifier, an ENPP1 mutation, for example. Here you see a pedigree with an arrowed patient who has 2 ABCC6 mutations. And this was a patient that I followed until the age of 40, 42, and everything was fine, and he didn't really have a lot of symptoms. And then over a period of 6 months, he developed extremely severe vascular disease. And you can see his images below of his blood vessels. If I would see that image and not know the patient or the pedigree, I would just say this is an 80-year-old patient. Extremely severe with severe artery disease, which is not on such short-term PXE, but he was found to carry also a genetic defect in the ENPP1 gene. And that ENPP1 gene is aggravating his vascular disease and made it so extreme, so rapidly progressive. So ENPP1 is really a very strong modifier of vascular problems in, for example, PXE. The VEGF gene variants are more linked to the eye disease and is much more subtle. There are 4 of them marked here in red that are associated with more severe eye disease, but more severe eye disease being defined as decreased vision or the need for anti-VEGF treatment, Avastin, Macugen, Lucentis, these anti-VEGF treatments. So we see that if you carry these variants, you can have a higher chance of needing these anti-VEGF treatment. It does not mean that you will completely lose your, in many cases, these treatments work fine and patients will retain their vision. But this is why in a diagnostic flow that we screened for several genes. Some of them I've discussed, C6, ENPP1, GDCX. There are 2 others that are novel ones that may be related to PXE to confirm the diagnosis and to enable carrier testing. And then we also test some modifier genes in one analysis, but this is done mostly in a research basis to allow prospective evaluation of the effect of these modifiers. And for example, we follow the patients who have these VEGF-A variants a bit more closely compared to others to monitor if their eye disease is really more severe and if we have to intervene more. And so to end up, so the diagnostic flow that we use for PXE is also been a bit modified based on the possibilities that we have now due to this genetic evolution. Of course, if we can, we will retain ourselves to the genes that are really linked to Pseudoxanthoma elasticum. And if we find the genetic defects using that, that is fine, and we stop. If we don't find genetic defects in these genes, we will go further and look at a number of genes, about 300, where we know that they are very crucial to Pseudoxanthoma elasticum. If that doesn't work, we'll look at all genes together and see if we can find anything. And then worst case scenario is a full genome analysis, but the number there is so difficult that as a clinical geneticist, this is sort of the horror story because the information that we will get is not really don't really use that in a clinical setting. So summarizing the genetics behind PXE, you have a first layer of disease-causing genes, mainly ABCC6. There are a couple of others that in a smaller subset of patients can be relevant. These actually cause the disease. Second, you have the modifier genes, the layer below that, that will not cause the disease, but that will progress and how it evolves, either large effect modifier genes or small effect modifier genes. There is a third layer that I haven't discussed now, which is called epigenetics, which will really do the fine-tuning of the disorder, and is sort of important for the interaction between your genetic material on the one hand and the environment on the other hand. And all these layers together will, in the end, lead to the things that we see and that you see, and that is the symptoms of Pseudoxanthoma elasticum. Of course, the ectopic mineralization, but more importantly, the skin, the eye, the cardiovascular, the gastrointestinal signs that we have. And with that, I will thank you all for your kind of virtual attention. And if you have questions, I will be happy to answer those.
Great. Thanks very much, Olivier. What we've been doing here is some people, let's see if they get your joke with your genetics mutations there. Some people have been putting questions in the chat. I'm going to make us back into gallery mode so that we can see if people raise their hand is the other way that people have been doing this. So we do have a couple questions already in the chat. So one is if stress is a part of the environmental impacts, and what do you believe stress has to do with, for example, vision loss and other aspects of PXE?
Yeah, well, I think that stress in itself, of course, doesn't cause a disease. But for example, it can be important in the occurrence of high blood pressure. And of course, high blood pressure is a separate risk factor for heart and vessel disease. And if you have the combination of that together with PXE, then that will influence or can influence, for example, your phenotype. But on skin disease or eye disease, stress itself will not really have a large effect. And in general, in medicine, you know, we always say that you have to avoid stress. That's easier said than done. But the impact of that is not that huge.
Okay, we have another question about whether or not you've seen P10, P-T-E-N, associated with PXE.
No. That's so you mean either Cowden disease or autism with a large head circumference due to P10 mutations. But this is coincidence, unfortunately. But I have never seen a patient with PXE and a Cowden disease. But we have to say that more frequently than before, we see individuals who are diagnosed with 2 genetic disorders. Because of course our diagnostic capabilities are becoming better, and so we are detecting a lot more, which means that given the fact that we all at least carry 5 genetic defects that are disease causing, that we will find more and more patients that have 2 genetic defects, but one not necessarily related to the other. And so the dominant P10 mutation has nothing to do with the recessive PXE mutations.
Great, thanks. And I often remind PXEers that that's true of common conditions as well. You can have PXE in arthritis, or PXE in breast cancer, or PXE in psoriasis. Unfortunately, we humans get a lot of different ways to have variation in our genes and in our environment.
Yeah,
other questions? Let's see. Looks like no more questions. Give it a second.
I have here, in case of the person who worsened within 6 months, were there recent environmental changes in his life? No. So that was a bit, that was the thing that at that time we did not check other genes. We just performed ABCC6 analysis for PXE, and that was it. So we didn't detect the ENPP1 mutation at that time, but that was one of the things that triggered us to look further, because it was so unexpected. There were absolutely no other cardiovascular risk factors in this patient to explain the rapid progression of that vessel disease.
Okay. I'll just give it another second, because sometimes it's hard to either unmute or raise your hand and see if there's anybody else.
Hello?
Yes.
I had the 100,000 genome study, which said that I was inconclusive, although I'd had PXE for 30 years, diagnosed clinically. And then after another year or so, they came back to me and said they'd looked at it again and decided that I did have it because I'd had 2 faulty genes. Why did they not find that before?
Yeah, okay. Well, first of all, certainly with PXE, very often it's not the molecular diagnosis that decides if you have it or you don't have it. I mean, there are other ways to do that, and the gold standard is still look at the symptoms and the skin biopsy. And in a lot of cases, based on just 2 STU, you can say you have PXE, and the clinical diagnosis is made. And then the second question is, of course, can we find the causal genetic defects? If we can't find them, that does not mean that you don't have the disease anymore. Because you still have the symptoms. Whether we find a genetic defect or not, you still have the symptoms, you still have the complaints. The disease had not disappeared with that. So if we do the genetic testing and we find variants, as I said, genetics is an evolving discipline. And you know, you learn time. And sometimes we find, let's say that we do the genetic defect in the genetic analysis in 2019. We find a genetic variant in your genetic material, and it's the first time that we find it. And then over a year or 2 years, we see other families, and we see that the same genetic variant we can find it in another family who also has a PXE patient. And we find it in a third family where this patient also has PXE. And so the evidence is slowly mounting up to say, okay, this is no longer, you know, the first time that we find this variant and we don't have a lot of information about it. We have more and more information slowly, and on the basis of a year or 2 years, we have sufficient information to say, okay, we have no doubt anymore, this is a genetic defect. And that is why very often, if we don't have a genetic diagnosis immediately in patients, not just for PXE, but in general, we say, you know, it is worthwhile to see you again in 2 years, in 3 years, and we have to look it up again. We have to re-evaluate again, and knowledge evolves, and we may be more certain about the meaning of a genetic defect in 3 years that we are today, because simply we have more information about that. So that is probably what happened in your case.
Yeah, because I went from PXE to presumed PXE, and then they were diagnosing my deteriorating eyesight with dry macular. So the treatment wasn't available.
With that conclusion is something that I don't completely agree with. If you have a clinical diagnosis, even when the genetic test is normal or not conclusive, you still have a clinical diagnosis. The disease stays the same. And I have to admit that I make a lot of clinical diagnoses, not just for PXE, but also for other diseases that we cannot confirm.
Yeah.
But where I'm absolutely sure, based on the set of symptoms that the patient has, that he has that disease. So the clinics is always the most important thing to make a diagnosis. It's different when you have symptoms that are not very clear, that are not typical or less typical for PXE. Then there may be doubt if the molecular testing is not conclusive. That's
my symptoms are very clear.
Yeah, in that case, I wouldn't doubt and make a definite clinical diagnosis and then see what comes out of the lab.
Thank you very much. Thank you. Anyway, I've been confirmed, so.
Great. All right. Well, I think that's it. And we're at the top of the hour. So perfectly done, Olivier. Thank you so much. Very clear. Yes. Thank you.
Okay.
Have a good evening.
Here. Enjoy the rest of the meeting. See you tomorrow for the Zebrafish pitch.
Wonderful. We'll look forward.
Bye-bye.
Bye-bye.
Okay.
Bye.
Bye.
Okay. So we are at the top of the hour, and we have on your schedule guided movement visualization break. And so Tamra is going to lead everyone in a short 15 minutes or so, maybe even a little less. So we also have time for water in, water out break before we go into our next session. You should feel free to follow along. You should feel free to walk around and do something different. We just want to make sure we're breaking up the time that you're spent staring at the computer and sitting, since I think we've heard smoking, sitting, these things are probably not the best for all of us. So Tamra,